For the invitation. And hello, everyone. So my name is Qiaobing Xu. I'm currently an associate professor in the Department of Biomedical Engineering at Tufts University. I also hold the affiliation with several other departments, like the Department of Chemical Engineering, and also hold the faculty position in Tufts Medical School. So today I'm going to talk about some of our work in the area of synthetic lipid nanoparticle for drug delivery. But since I will also provide an overview of the nanomedicine, considering many of the audience may not have may not work in the same area, so that it's pretty general and we'll see, before we go into the science, so I want to talk a little bit about Tufts University. So Tufts University is in Massachusetts. So Massachusetts is a very small, can you see my pointer, the mouse? Can you see my mouse?
No, we cannot see your mouse.
Okay, so maybe it should be in this way. Let me see. Can you see here?
Yes, perfect.
Okay, so yeah, Massachusetts is a small state in United States and in East Coast. And Tufts, we have three campus. The main campus mainly for graduate students and undergrads. It's in the suburb area of Boston, the capital of Massachusetts. So I'm in the School of Engineering. So actually we are in the main campus in the Medford and Somerville area. And in downtown Boston, we do have another campus, which is our Tufts Medical School. So we have the research there and also the hospital there. And the hospital currently that they see a lot of patient COVID-19 patient in our hospital. So that's in the Boston area. About 40 miles from the downtown Boston. So we have a veterinary school in Grafton. So there they have hospital to see the animals like horses, dogs, and pets. And there are also lots of the research in there as well. So including the infectious disease as well. So basically, Tufts has all both graduate program like PhD, masters, and also have the undergraduate program. So our undergraduate program are pretty good. So it's always ranked from 27 to number 30, like each year from all university ranking in US. So this is a little bit about the overview of the Tufts, just in case that you probably don't know. So let's get into the science. And my main research area is in the area of the nanomedicine. So while for the nanomedicine and so in other words, so drug delivery. And here it showed the length scale of different subjects. So like the size of the tennis ball is about a few tens of the like 10 centimeter in the diameter. For the cells, so like a mammalian cell in our body, the size is about 10 to 40 micrometer size, depends on the cell types. And some other objects that we can find in the cell or sometimes, so like the virus, COVID-19 is a virus. So the size is about, depends on the type of size of the virus. So it's about a 40 nanometer to 100 nanometer, depends on the type of the virus. And there are also the molecules in the cells or the virus like DNA, proteins, small molecules. So those are a few nanometers or sub-nanometer scale, right? So why we need to actually make nanoscale medicine in order to treat the disease, right? So the scale is very important because you see that the cell is about 10 to 14 micrometer size. while in the cell, so there are a lot of the organelles that's around like a few nanometer size. So if you want to actually treat or fix the cell, so you'd better to have something that's in that scale, right? So that's why the nanometer size objects can actually treat the disease in the cell. Okay, and while you for some of the stuff that you want to get into the cells, right? So that carrier, for example, whether it's a virus or non-viral carrier, so the size has to be a few nanometer or a few tenths of the nanometer scale. So in order to get into the cell to fix the stuff that you want to fix, right? So that's why the nano is important in terms of the therapy or treatment of the disease. Okay. So what we showed here, there's a different type of the nanomaterials, and if you work in this area, so you may already know like golden nanoparticles, quantum dots, the polymer nanoparticles, and some other nanomaterials are based on the self-assembled nanomaterials like the micelles. So actually the molecule itself is a small molecule, or the liposomes. So, but they can self-assemble into like vesicles, micelles. So the size of those things could be a few tens of nanometer to a few hundred nanometer. So you can use those nanomaterials as carriers for drugs, and also you can use them for imaging and for sensing for different applications, depends on your research of the area. And while in my group, we are particularly interested in utilizing those things for drug carriers to deliver the drug into the cells. And so a little bit of background about the nanomedicine. So this is the field of the like nanomedicine or nano area. So it's mainly like used for example, monitoring, control, construction, repair, and the human body, right, to improve the function of our body. And so this is even though this is some kind of like sci-fi level of the ideas. So for example, if you can actually generate a nano machine, so the nano machine can actually get into the blood vessel. So for people who has high like a lipid deposition on the blood vessel, so that so then those nano machine can actually remove those plaque and then to clean the blood vessel so that the patient will have lower cardiovascular disease or potentially, right? Or for people who has Alzheimer's disease and because of the loss of the neurons, and if you can actually create nano-sized cells and then to connect those neuron connections so that may be useful to treat certain degenerative disease. So even though that those a little bit a little bit far from the reality, but actually I think that so with the progress of the nano medicine and also the nanotechnology area, so some of those things are approaching. So and related to the nanomedicine, so there already is some application of the nanomedicine for cancer therapy. So like this is like a blood vessel and some cell types like red blood cells and macrophage or monocytes in the blood vessel, and while in the cancer area, so that this is like a tumor. So when the tumor grows, and they have a defect in terms of the blood vessel formation. So those blood vessels are considered leaky because the tumor cell grow too fast, so that the blood vessel are not as well as good as the normal blood vessel you find in, for example, like lung, spleen, or not spleen, like heart. So those blood vessels, they are considered leaky. And if you have some object flow in the blood vessel, and for example, nanoscale particles, right? So those objects can actually get leaked into those like tumor area, and then they will retain there. So if you put some chemotherapy drug in the nanoparticle, and the chemotherapy drug will specifically accumulate in the tumor area, but not in other parts of the body. Oh, so that's called the enhanced permeability retention effect, so-called EPR effect. So actually, scientist and engineer has applied this effect and then to specifically to deliver the drug in the tumor area and not to the unwanted area. So to minimize, for example, the side effects. So this is one particular application that already get used to treat the patient with the cancers. So for the nanomedicine, and in addition to the like the vesicles I mentioned, so there are many other type of the subjects like the polymer nanospheres, the micelles, and the dendrimers or other polymers, other inorganic materials as well. So the general idea in the nanomedicine is you have various way to encapsulate whatever you want to encapsulate, whether it's a small molecule drug or biologics, and then so that will form the nanoparticle. And the nanoparticle will be uptaken by the cells. So when they actually reach to the cell, and the cell will uptake those nanoparticles mostly through the endocytosis. And if you have some way, so the nanoparticle will actually escape the endosomes and release the drug in the cytoplasm. So it depends on the type of the drug. So for small molecule-based drug, those drug doesn't have to actually escape from the endosome, so they will diffuse out themselves. But for large molecules like the nucleic acid, mRNA, those things, they have to escape from the endosome. Otherwise, they will get degraded in the endosome and they will lose their biological function. Okay, so I will give you some example later. So this is the general how the and some successful story in the nanomedicine. So like Abraxane is one of the nanomedicine that's approved for treat like breast cancer. So in this case, so they have the nanocarrier is actually an albumin, like serum albumin, and the drug like paclitaxel is a small molecule. So it's actually binding to the hydrophobic cavity of the albumin. So the overall size of this protein binding the drug is about a few nanometers. So while serum albumin is very abundant in our human body, so that it's native and it won't to cause some side effects. And they showed that this drug can have lower side effects comparing with traditional paclitaxel, this small molecule drug. Okay, so this is one of the examples. And another example is using the liposome formulation to formulate this chemotherapy drug called doxorubicin. So while doxorubicin, this drug, if you administer intravenously, so they can actually cause a heart attack or cause a heart injury because those side effects. And engineers or scientists have actually encapsulated this drug in the liposome formulation and then using probably like a EPI effect to deliver this into the like tumors, and then so that they can minimize the cardiovascular the cardiotoxicity of this drug and to make it local to the cancers. So those are some examples of the liposome. So since my talk will be particularly related to the liposome-based drug delivery system, so I want to give you a little bit of background about the liposomes and the formation of the liposomes, just in case that you don't have the background. So as we know that the cell membrane is actually made of those phospholipid, right? So the phospholipid are amphiphilic molecules, so they have hydrophilic head and hydrophobic Yankee chain, so they can self-assemble to form these bilayers. So if you throw those, it's like a soap, okay? So if you throw this molecule in aqueous solution, so they can form these vesicles. So inside of those vesicles are hydrophilic, and while the shell of those vesicles are hydrophobic because this is made by the lipid tail, right? So if you have a drug, for example, like a doxorubicin or other type of drug like paclitaxel, if you want to use this to encapsulate the drug, so mostly the drug will be encapsulated in the shell if the drug molecule is hydrophobic, right? But if it's a hydrophilic molecule, so they will be encapsulated in the cavity, like hydrophilic area, right? So the liposomes, the toxo, the example that I showed you, so they have been used to treat cancer patients. So they have some features. For example, they can increase the solubility of the drug. They can increase the stability of the drug. They can lower the irritation, and you can also do a lot of changing the formulation to change the size by changing the lipid composition. A lot of the engineer side on the liposomes. Okay. So in addition to the toxos that I showed you, so actually liposomes has been used for many other drugs as well, including the antibacterial, antiviral, and the fungicides vaccines as well. So using the liposome formulations, so many of those are in the clinical trial or even approved. For example, like this one called amphotericin. So amphotericin here. So this is also approved to treat the fungi. Okay. So the example that what I showed here, the drug molecules are all small molecule based drug. So while with the advancement of the molecular biology, so we got to know more about the root of the certain disease. So there are new therapy that has been created to treat those disease from the root rather than from the symptom. So that gets into the gene therapy or nucleic acid based therapy. So this scheme showed the central dogma of the molecular biology. So we know that for the cell or for the life bodies, so it will we have the DNA, which is genetically encoded information in the DNA. And while the DNA can be transcribed into the messenger RNA can be translated into proteins. So the protein is the functional one, while the DNA sequence determine what protein will make, right? For people who has, for example, genetic disorders, if they have the mutation in the DNA sequence, so they will make the wrong protein, and then the wrong protein will not be functional, right? For those type of the disease, and you can imagine the treatment will be whether you can actually correct the mutation in the DNA lab, or whether you can actually put a functional mRNA into the cell, right? Or whether you can put a functional protein into the cell, to restore the function of the cell, right? So that's sort of the novel therapy in the gene therapy area. Okay, so how do you actually get those, for example, like apparatus to get into the cell to correct the mutation? Or how do you put the mRNA, the functional mRNA, into the cell to make the functional protein? Or even how do you put the protein directly into the cell, right? So that's a delivery issue, right? So and while for the gene therapy, this is an example, so like called a cystic fibrosis. And while for this particular disease, it's due to mutation of the ion channel of the cell membrane. This is chloride ion channel. So due to the mutation, so the chloride cannot actually pump through this channel, and then they can accumulate the mucus on the surface, and then that lead to the infection and also the damage of the tissue. Okay, so one of the treatment is whether you can actually put a functional chloride ion channel on the cell membrane, right? So that will make the cell function. So the question, is how will you put this back, right, into the patient? So that's a delivery issue. So and while for the gene therapy or nucleic acid based therapy, so this is like Many are in the clinical trial. So in terms of the delivery of the gene or nucleic acid, so viral-based are commonly used using the virus or engineered virus. So for example, like adeno-associated virus, retrovirus, or adenovirus, HSV. So this is like the cases of the gene therapy in the clinical trial. So actually, you will see the majority are using the virus-based, and they are a small portion. So actually, using the naked plasmid DNA or using the like liquefaction. So liquefaction is using the lipid. So this is the statistic about the gene therapy. So this the cargo here is to deliver the DNA. Okay, there is an inherent purpose why the virus is particularly good for the gene therapy. But as I mentioned here, so in terms of, in addition to deliver the gene for the gene therapy, there are also other options so that you can use a functional protein or you can use using a functional mRNA or protein, right? So that or even use an RNAi, like short interference RNA to knock down certain genes. But while for those things, so viruses are not as good as some of the non-viral approaches, so like including the liposomes, polymers, and the other approaches. So while my particular research is in the non-viral area, so for the non-viral area, in particular, the cationic lipid are commonly used for the drug delivery, including for the delivery of the nucleic acid. So those are cationic lipids. So it means that those lipids are positive charged. So those lipids are very different than the lipid that I mentioned before to form the liposomes. So the liposomes from our native like cell membrane or usually negative charge, while this particular lipid are synthetic, so they are positive charge. There is a purpose to make this kind of positive charge lipid because the cargo, like DNA, messenger RNA, or sRNA, or nucleic acid, right, are all negative charge. So the positive and negative charge can actually self-sample into through the electrostatic interaction to form those nanoparticles or the multilamellar vesicles. So this is the cryo-TEM of those multilamellar structure. So this is through the interaction of the negative charge of DNA and also positive cationic lipid. Okay, so this is an example to use this lipid to deliver green fluorescence protein plasmid into the cells. So the cell will make the green fluorescent protein and they will turn into green. Okay, so that's and while for this particular lipid called the lipectamine, so it's commercially available from company like Thermo Fisher, and it's pretty popular in the research. And while for the lipid itself, it's pretty toxic, and it cannot be used for the human use because of the toxicity, even though it's pretty effective in vitro. So there is a desire to develop the new lipid that can deliver, for example, the DNA, RNA for therapeutic application, right? So actually last year, there is a breakthrough in this area so that there is a first like FDA approved synthetic lipid-based nanoparticles called Onpattro. This is the lipid used in their formulation. So this lipid this lipid is used to deliver an sRNA, and that can actually knock down a protein called a TTR. So that this lipid sRNA combination called patisiran or Onpattro, which is made by Alnylam, a pharmaceutical company in Cambridge, Massachusetts, so not far from here. And so to treat the patient with the TTR deficiency to this. Okay, so this is like a big progress in this area in terms of using the synthetic lipid, right? But while this is only one, and also this is only good for the live delivery, so there are a lot of other diseases whether you can actually engineer a good delivery system to deliver the desired cargo, whether it's mRNA or the siRNA or DNA into. So there's a lot of need to make these kind of molecules, right? So at Tufts, in my group, and we're using the complementary library strategy to make those synthetic lipids. So one of the lipid that we made, those cationic lipids, so they have a disulfide bond and they have a positive charge. The tail is aliphatic, right? So that those are the cationic lipids. So comparing with what that has been developed before, so we include a disulfide bond in this class. So the disulfide bond is cleavable inside the cell so that to make this lipid degradable. And we hypothesize that due to the additional degradability, so those lipids can actually be more biocompatible. And while the chemistry is very simple, so it's based on the microaddition of the primary or the secondary amine and with acrylamide or acrylamide. So it's just this put those two reacting together, heat them, and then we can make a lipid-like molecules so that we can change the type of the amine, for example here, and the type of the tail, and then to make the combination so that we can generate a library of those molecules. So then we can do the library screening and then to do use this to do the delivery. So this is like a transmission electron microscope to show the one particular, the morphology of the self-assembly of one particular lipid. So it's like worm-like structures. So what we usually, how do we screen if we make thousands of those lipids, right? So we usually do in vitro screening first. For example, we check the efficiency if our cargo is for like knockdown, and we check the knockdown efficiency, and we also check their toxicity, which is in the cell level, like cell viability. We'll also check the hemocompatibility to check their hemolysis capability. So what we want is something from the library in this corner. So that means that their knockdown efficiency is high, their cell viability is high, and their hemolysis is low. Right? So those are the like a best candidate to move into the in vivo, at least from the in vitro. So that's sort of what we, how do we do the screening? Okay. So based on this strategy, we also made a few other variations. So for example, in this case, we changed this to heteroatom other than the carbon. So we include a single oxygen, selenide, and sulfur, and selenide, and to change the physical property. And using the chemistry, we can generate a new library. And in this case, we can change this to the acrylamide and to make a new library. In this case, we make our lipid to be photosensitive. So this is a photodegradable link. So after shining the UV light, they can break. And in this case, we change the head to like try acrylic acid. So for this lipid, they can bind to the metals and then they can add the new functions. So we made a lot of the variation in terms of the chemistry. So those are all published. If you're interested, you can read those papers. And as in the next, I will show you some examples that we have done using the system. So this is sort of the general, the overview, what we do in the lab. So this is a library, and then we do the screening, depends on the cargo, could be protein, peptide, protein nucleic acid complex, and nucleic acid, or even small molecule, depends on the cargo of interest. We do run the screening, and then we sort of do, even though that we didn't do like a human study in the lab, we do the study in the animal. But using the screening, we can actually identify the lipid. The nanoparticle can actually get into different organs, for example, the lung, liver, spleen, and the intestine and the brain. So that it's pretty interesting system. Okay, so some examples. So what I showed you here is an example. We screened our library, And then we showed that it depends on the nanoparticle types, so that we can deliver, for example, the messenger RNA into the liver pretty specifically. So this is the luciferase mRNA, and we can image that using the in vivo image system. So anywhere that express luciferase, so that will turn on. But for the place that they don't have the luciferase, and they will they don't have the signal. So this is pretty convenient for us to study where the luciferase mRNA get, right? So for this case, they get into the liver, and they get into the lung, and they get into a majority to the spleen. So the interesting part here, you can screen the library and then get into like the areas or the organs that is interesting. So with one particular example, so that we used our delivery system to deliver Cas9 mRNA together with a guide RNA and into the liver to knock down a gene of interest called PCSK9. So PCSK9 is a gene that's related to the cholesterol level in the body. So if we knock down the PCSK9, so that will actually can lower the cholesterol level in the liver in the body. So this is a valid target to treat, for example, hyperglycemia. And what we showed is we actually deliver the CRISPR-Cas9, the enzyme, the mRNA, together with the guide RNA that target the PCSK, so that in this paper we identified this lipid, synthetic lipid, so they can actually deliver the guide RNA and the mRNA into the liver and to knock down the PCSK down to like 20%. So this is very, very effective in terms of gene silence using the CRISPR-based approach. And we also checked the toxicity of the delivery system, so that you see that this is like histology. So comparing with the PBS, the treated, so there is no damage in the liver. So we expect that this formulation can actually potentially can move into the clinical to treat patient with hyperglycemia. So this is another example that we used our bioreducible lipids to deliver the antisense oligonucleotide. So this is the small nucleotide, single chain. And again, in this example, we used PCSK as a model. So what we showed is we can actually load this SO into our cationic lipid, so they can get into the cells effective. So in this paper, you can read more. And we have studied screening the library in the cell level, for example, to use the GFP as a model. So that will allow us to identify an effective lipid. And then we use the top lipid screened from the in vitro, and then move into the in vivo. So in the in vivo, what I showed you here are just some in vivo data. So actually here, what you see, those are the organs from mice. So we tested the 3 top lipids that were identified from the in vitro screening. Excuse me. So in this case, we delivered the near-infrared dye labeled ASO, and then to check the biodistribution. So what you see, this lipid can actually get into the liver more effectively than other 2, right? So, and also we checked the PCSK level, the knockdown efficiency of this particular formulation. So we showed that the ED50 is about 0.03 or 0.09 milligram per kilogram. So this is sort of the best data shown in literature comparing with, for example, like siRNA data from the NLM. So this again showed the efficiency of our lipid. So what I showed you, the two examples, this one is the antisense oligonucleotide, so it's a nucleic acid as a cargo, and this one is the messenger RNA as a cargo. So both are nucleic acid, they are naturally negative charge, so they can actually form the complex with our cationic lipids through electrostatic interaction, right? Um, another example that I want to show you that using our lipid, we can also deliver the protein for cancer therapy. So while the proteins, it's made of the amino acid rather than the nucleic acid. So amino acid, so actually the protein are not necessary to be the positive charge, but the protein has a negative, not necessary to be negative charge, but a protein has negative charge, like a moiety, and also they have the hydrophobic cavity, so that can actually form the complex with cationic lipid. By no means we tried in this study, we tried to deliver two proteins. One is saccharin, and the other one is called ribonuclease A. So for saccharin is a toxin. So it's actually a protein isolate from the seeds of a plant called Saponaria officinalis. So it's a ribosome inhibitor. It can inhibit the ribosomes, so potentially can inhibit the protein synthesis. And while it's not toxic if it's outside of the cell, because itself cannot penetrate into the cell. But if you have a way to get this into the cell, they can actually induce, for example, the ribosome inhibition, and then induce the cell apoptosis, right? So if you can get this into cancer cells, so that can actually activate cancer cell, that can be useful as a cancer treatment, right? And while ribonuclease A, which is like RNAs, again, for this protein, if you introduce into the cell, they can degrade the RNA and lead the cell to the apoptosis. But if it's outside of the cell, it's not toxic. So that's why we want to deliver those. And also both of those proteins are in the clinical trial for cancer treatment. So we believe that an effective delivery system can actually improve the therapeutic efficacy of those proteins. So we tested our library. So we use the cell viability. So in this case, we use our lipid to complex with those cargoes, and then add into the cells. So those cells are cancer cells. So if there is no uptake, then the cell will survive, right? If there is a cell uptake, then the cell will die. So we can check the cell viability which can predict or tell us the efficacy in terms of intracellular delivery. So what we showed here is, for example, this lipid, they can effectively deliver, for example, separate into the cells, lead to the cell toxicity, right? And then we tried in different cell types, so including the breast cancer, melanoma, liver, prostate, and the cervix. So, and using our delivery, they all showed pretty good the IC50, which is like 50 times lower than the protein alone. And you can imagine using our nanoparticle to deliver this protein. So when we use this to treat the patient, the dose will be 50 times lower than what's currently used in clinical, right? So that will be a huge benefit in terms both of the cost and also the compatibility. And we in this particular paper, we also showed that our nanoparticle can deliver the saccharin not only in the cell level, but also we can actually treat the tumors in the mice model. So this is the experimental procedure. So in the day one, this is like a 41 breast cancer syngenic model. So what it means 41 is breast cancer cell types that derive from the mice. Okay, this strain, and we culture in the lab, and then you inject into the mammal fat. And then after the injection, so the cell will survive and around a week or two, so they will form tumors in the breast area. Okay, so and after the tumor form, so what we did is using like IV inject the nanoparticle complex with saccharin. So using the IV injection, so we did a 3 treatment. All of them are through the intravenous injections through the tail vein injection. So we monitor we monitor the tumor size. So we find that the tumor treated with the lipid and the saccharin complex can suppress the tumor growth, while in carrier alone, like EC16-1, or the saccharin alone, there is no such inhibition in the tumor size, right? So this shows that we can actually using this lipid so that we can potentially deliver the saccharin to the tumor cells and then suppress the tumor cell growth. So this is the histology to show that the protein delivered by the lipid nanocomplex do get accumulated in the tumor area. So this is the staining of the protein, because this protein is not found in anywhere in our body or in mice body, because it's exogenous, so that you can actually very easily using an antibody against the saccharin to stain this protein. So you see that compared with the protein alone and the protein complex with the nanoparticle, the accumulation in the tumor area more significant. Right? So this is one of the evidence to show that the observation that we show here is actually due to the delivery of the saccharin in the tumor areas by these lipid nanoparticles. So here we showed an example to deliver the proteins. So in this case, we use our lipid to deliver like a fusion protein. In this case, it's called the pre-recombinase, fused with GFP. So when we deliver this protein to the cells, it can actually remove a LOXP gene in the cell and turn on the protein production of the downstream, which is red fluorescence. So we can monitor the red fluorescence, the population of the red fluorescence cell that can tell us how effective our delivery system are. So we run the screening. So you see that this is we test like 12 comparing with the lifectomy, which is the lipid from the in vitro gene. And many of our lipids showed more effective than the lifectomy in terms of delivering these proteins, right, in the cell lab. And we also test this in the mice. So this is in the intrabrain injection. So we inject those nanocomplex in the AI14 mice, and you see lots of the red dots. Red dots in those mice treated with lipid complex with the GIP3 proteins, and comparing with GIP3 protein alone, you don't see anything. So this indicates that the protein alone cannot get into the cells. And while using our lipid, they can get into the cells and then successfully to do the gene recombination and turn on the TT tomato, the red fluorescence gene in those cells. So we can expect that using this kind of delivery and loaded with functional cargoes, so this can be potentially useful to treat many neurologic diseases, including the Parkinson's or other type of disease. So, and finally, that I want to, I forgot to add a summary. So in the summary, so in the first part, I sort of give an overview of the nanomedicine and in terms of the small molecule delivery. And the second part, my main focus is in the nucleic acid delivery, protein delivery, right? So that's sort of the new version of the drug comparing with the traditional small molecule based drug. And while in the last part, so where I talked about some of our ongoing research in terms of developing the lipid-based nanoparticles to deliver the nucleic acid, including the antisense oligos, mRNA for the gene editing, and also the proteins for cancer therapy and also the gene editing. So that's sort of something that I covered. And finally, that I want to acknowledge, so my students, my group, and also my collaborators. Lastly, I want to talk a little bit about our Department of Biomedical Engineering in Tufts School of Engineering. So we have BS degree in the college for the undergrads, and we also have the graduate level, like masters and also the PhDs, and also certificate for in the biomedical engineering. So in our departments, we have mainly three main research areas: the biomaterials, including people working with tissue engineering, tissue models, and my research area falls into the biomaterials, but in the drug delivery area. There are two other areas, including the biomedical devices and also optical imaging. So those are the three main focus of our department. So that you're welcome to check our website and find the faculty and the interest. So if you are interested in applying the graduate program, so feel free to submit your application. So finally, I want to thank you and also answer any question that you may have.
Thank you so much, Qiaobing. Thank you so much. Excellent project and chemistry. Really, really interesting. Um, there are a few questions on the chat and a few others. Well, maybe we can start. First, do you have any advices for students who would apply for a master or PhD in your department? What do you see the most important part? Is it the recommendation letters? Is it the publication? What is the key for students, to get accepted in, for example, in your biomaterials program for master or PhD?
Yeah, in in our department, so we all the faculty are sort of in the review committee for the graduate applications. So usually the applicants will indicate their like a research area or potential faculty advisor in terms of number one, two, three, and then those faculty will review their applications. And so what area, what I would say that all matters, including the resume, the research experiences, and um, for undergrads, it's hard to expect that they already have the publication, right? So, but with some research experience will definitely be helpful. So, I would recommend the students who can or who has a capability to do some research during their study, and so that will be definitely a plus.
Excellent. Thank you so much. So there are a few questions. I don't know if you can.
Uh, so the question is, can you please comment how do you integrate the team of the nanomedicine with the synthetic team? Oh, integrate. Oh, you mean that my team? Okay, so I guess that you say that in my research, we have someone work on the synthetic chemistry and someone work on the cell and also the mice work. So actually, a lot of the work that I showed you from my group, so most people can actually start from the beginning to make the new materials and formulate them and do the animal work to the end by uh, each of the students. So after the training, they can actually do all those things. But now my group is getting bigger, so I have some student who is very good at chemical synthesis, but have little biology background, molecular biology. So I have student to do very difficult chemistry, and that will cost a lot of time. And I have student making those messenger RNAs, so they will train as a molecular biologist. I have student to make the formulation and test the animals. So it will be a teamwork in those cases. For the teamwork, depends on uh, who initiated that project, right? So if, for example, the messenger RNA or RNA is a key factor in that project, so probably that person who make the RNA will lead to the project and ask others to help to do the formulation and do the study. And later, for that student, he will also learn the animal things. I think that he later will get trained and do it by himself. So that's sort of how my team worked. And so the next question is, what are the common side effects of the liposome drug delivery on patients? And can any drug be liposomal encapsulated, or are there specific limitations given by the drug? Very good question. So can let's answer the second question. Can any drug be encapsulated using the liposomes, right? So theoretically, yes, because for the liposomes, they have the hydrophobic shell, which can encapsulate hydrophobic drug, and they have the hydrophilic cavity to encapsulate hydrophilic drug, right? But in reality, some drugs are not easy to encapsulate. For example, like um, aspirin. So aspirin is a hydrophilic drug. It's a very water soluble and small molecule. If you even you can encapsulate it in the cavity, but the leaky, so it's not that stable. So that the formulation that you make may not be stable enough to actually for the practical application. Okay. So it will be case by case, and how will you formulate those liposomes? So the question, are there specific limitation given by the drug or the liposomes? Are there? Yeah. So for the liposomes, it's definitely that not say that the even though that's it's a good delivery system, because you see that many of the drug in the clinical or approved that's using the liposome formulation, but there are also limitations. For example, like stability of the liposome storage and the skill used to make those liposome formulations, or could be a challenge, could be a limitation for liposome-based drug delivery system. And the first question, what are the common side effects of the liposome drug delivery on the patient? So the side effects, that depends if it is using the traditional like a lipid. So the lipid itself is not very toxic because it's made of the cell membrane you can find from the nature. So in that case, they're not too much toxicity. Uh, well, for the cationic lipid, so depends on the dose, potentially that they can cause some side effects. For example, the liver toxicity, or depend on the dose. So that's why in one of the data that I showed you, we did the liver histochemistry to check the liver toxicity, right? To make sure that the dose is right in terms of with minimal or no side effects. But if you do inject tons of those things in the body, and that can cause toxicity. So the other question, what is the trend on the use of principle of polymer synthesis, especially ionic polymers, to generate the type of the material you present today? Oh yeah. So even though that my research are particularly focusing on the lipid, right? So while there are tons of excellent researchers in the world, they're working on the polymer-based delivery system. So actually, many excellent polymer-based drug delivery system that has been developed, published in the literature. You will see, and some of those are actually uh, maybe in the clinical trial as well. So one of the prominent researchers in this field is the Kataoka professor in Japan. So he's one of the leader, the polymer based delivery system. So it depends. I think that hopefully that I'm not trying to get you biased that the lipid is better than the pollen, right? So if you're interested in the pollen, so certainly that you should explore that area as well. So that's actually my own training is a polymer background. The specific accumulation in tissue is due to the functional group on the surface. Um, so this is an excellent question. So that the accumulation in the tissue, right, or tissue targeting. Yes. So even though that we don't have the direct evidence, but we expect that it's due to the surface chemistry of the LNP because there's nothing else there other than the difference in terms of the charge and the hydrophobicity of the nanoparticle that lead to the different organ targeting. So in case we would like to use those liposome, in case we would like to use those liposome drug for cancer treatment application, can we take advantage of the EPI effect? Why do you think it has not been successful when coming to the real cancer patient? So this is a very complex. This is a question related to the EPR, right? So EPR effects. This was proposed by Rakesh Jain from the MGH and many other, quite a few others. So definitely that there are some effect, or people see this quite often in terms of the delivery in the cancer environment, and why. It's not has not been very successful to treat the real patient, right? So I would say that because the cancer is very heterogeneous, and also the cause of the cancer and the cancer microphysiology is very complicated. So just the relying on the EPI effect is not enough. Even you know that um, so so currently that there are a lot of combination therapy to use chemo and also immunotherapy. Even they find that say that a immunotherapy that works for one type of patient, but they don't work for all, right? So that is show the complexity of the cancer. So even though that they are the EPI effect, I would bet that if you check the 10 patients, the EPR is not all the same, right? So uh, it definitely is it definitely is there, but how successful it could be, that's a question. So you can read a paper by Professor Warren Chen in University of Toronto about the EPI effect. So he wrote a Nature Review paper, very good review paper talking about the EPR. So the question that is so low solubility, lack of the stability is regarding the liposome, how to manage it. Oh yeah, low solubility. That means the low solubility of the drug. So the drug, if it's a low solubility and it's hard to formulate, but you can use the liposome to encapsulate to increase its solubility, right? So that can solve the formulation issue. Lack of the stability that refers to the liposome formulation, because the liposome is formulated is through the self-assembly, so that it's thermodynamic process, so it can actually, the drug can get leaked compared with the polymer encapsulation, polymer delivery system. So the lack of the stability is one of the formulation challenge when you work with the lipid. So are your LNP susceptible to be degraded by the phospholipid A2? We haven't checked that. So we have acid bond. So the disulfide bond definitely cannot be degraded by these enzymes. The phospholipid, the acid bond may be, but it's not a natural lipid. So I would say that it's probably not the substrate of this enzyme. But we didn't try. Do you see opportunity with natural lipids, especially from plants? For example, there are significant limitations. Absolutely, I think that this would be a lot of the opportunity in the natural lipids, right? So actually, many of the liposome formulation they use the natural lipids. So, for example, from the soybean or other sources, and so they have the advantage of, for example, like low cost and abundant. So I think that would be actually a good research area. But for those natural lipids, they can only be useful to encapsulate or formulate the small molecule-based drug. So they cannot be very useful for the nucleic acid delivery. If they can, the efficiency would be very low. Okay, so that's the last question.
Yeah, Qiaobing, excellent. A lot of questions, a lot of good discussion.
Oh, excellent questions. Very good.
I'm glad that we were able to have you for a seminar. And Luke, thank you so much again, and we keep in touch. Always enjoy reading your papers. Really nice chemistry, really exciting things you're doing. So hope to catch up soon, Qiaobing. You take care, my friend.
Thank you, Frank. Thank you all for the excellent questions, for being here with me. Okay, thank you.
You take care. Bye bye.
Stay safe.